In this video, I will describe the multispectral microscope tool that has been developed through the AFSIN 2015 by Aboma Merdeza, Alvarez Cosono, and myself. So this is a general view of the microscope. We will start with a description of the light sources. So we have a RGB laser, so it's a 3 laser system with a red laser at 650 nanometers, a green laser at 532 nanometer, and a blue laser at 450 nanometers. Then we have an additional laser, which call it the violet laser at 405 nanometer. So those lasers are actually powered using a single power supply. In this case, it's simply 12 volt DC and 2 amps. So we connect it and we got light getting out of one of the lasers. So let's follow the path, the optical path. First, we control the laser power using a wheel containing neutral density filters. For each numbers correspond to a filter where the transmission change. You can actually see exactly the corresponding transmission for each neutral density filter you want. Then we got a lens that will focus the light into the optical fiber. This lens is an achromatic doublet spherical lens with a focal of 60 millimeter and 1 inch diameter. This is a diagram showing the difference between an achromatic lens and a normal lens. The light goes through the optical fiber, and those fibers have a numerical aperture of 0.39 and a band radius of 50 millimeters, so do not bend it too much. This is a diagram showing what a numerical aperture, how it is defined. The light gets out, and you can use a diffuser just at the output to have an homogeneous illumination and reduce speckles effect. Here we have another lens to collimate the beam. And this lens is exactly the same as the one used before, 60 millimeter focal and 25 millimeter diameter. Then you can use additional lenses. This is the transmission mode. So light gets transmitted through the system and reach the sample. Then we have another holder where we can fix any objective we want. The light crosses a fluorescing filter wheel where you can actually put any filters you want to have a selective detection and remove the illumination light. At the end of the rail you can fix a camera. In this case, we've got a specific camera built by Luminera with 2048 times 1088 pixels of 5.5 microns in size. Then let's switch. This was the transmission mode. Let's switch the fibers into the reflection mode. To do that, you should turn off the laser when manipulating the optical fiber. Now we are in a reflection mode. In this case, the light, we want to collimate the light using a collimating lens. In this case, it's an achromatic doublet again, but the focal is 30 millimeters. It's a shorter focal because we don't want to expand the beam too much. Then the beam cross the beam split, the light cross the beam splitter. It's a Q beam splitter to avoid ghosting effect. And here the characterization of this 50-50 beam splitter. The light reaches sample, and so we illuminate the sample, and all light that is fluorescing back or reflected from the sample is reaching now the camera. In this case, the camera can actually be moved along the rail if this is required. Then we've got 3 controllers units. Those controllers provide the local and computerized control of each single motor axis. So they are connected to 3 stages, as you can see here. Those micrometric stages have 25 millimeters travel range with 0.05 microns resolution in displacement as the shortest displacement you can do. Then you can mount different objectives, and you've got reflective objectives that are made of mirrors or standard microscope objectives made of successive achromatic lenses. This is an example of the reflective objective. It's a pretty large objective, so you will have to create space to mount it inside your system. The standard objective is a bit smaller, and here's an example. So the standard objective, you mount it on a small mount, and the reflective objective that is bigger on a bigger mount. Then when you move, you should move the stage manually in the 3 directions to verify that the sample doesn't hit the objectives. This is very important before measurements. Put the objective and you move it in 3. Then we have to control the lasers. And this is done using a data acquisition card from national instruments. Those are the specifications of this card. So this card helps sending 5 volts into the laser, and you can switch on and off the lasers with your computer. So you have to connect the wires at the right port of the national instrument cards. Then you switch on your computer, open MATLAB, and we got a MATLAB interface able to control both the cameras and the traverse. So here is how the software looks like. And you can move the stages, you can illuminate it. For example, here we can start the red lasers on a reflection mode. So we just activated it, as you can see in this example. You can notice the spot of light on the right corner because it's 50% of the light cross there. Then the collimated beam reach the sample and you image through the camera. Then you can, second example, we can start the blue laser on the transmission mode. So here's the blue laser, so the transmission mode. And what we want now is not to saturate the camera. So you need to start with a small exposure time. So you increase slightly the exposure time. By default, the exposure time goes to 0. And then you can see this red dot on the image. It means you are saturating the camera. This red spot indicates to you that you have too much light on your sensors. So you have to reduce the exposure time or reduce laser power, not to saturate. So now we do not saturate. So once we have a good signal and we do not saturate, we try to find the focus. And for that, we have a built-in function called find focus that you activate, and it moves the sample until we find the focus. And now we get it. So 6 multispectral microscope 2 have been constructed for Ivory Coast, Kenya, Burkina Faso, Ghana, Mali, and Senegal. 6 projects are initiated with results submitted to the AFSIN board by November 2016. Then a minimum of 1 research journal publication per microscope is expected by the next AFSIN workshop in 2017. So with that, I would like to thank you for your attention.